Once all the reagents and supplies are available, now we can proceed further for PCR amplification. For this purpose, the first step is to mix each and every required material or supply in a specific order. For this purpose, we have to use a sterilized centrifuge tube or sterilized microfuge tube and add the reagents in the given sequence. First of all, we have to add the amplification buffer, the required volume, then the building blocks, the DNTPs, forward primer, reverse primer, thermostable enzyme, water, template DNA, and making the volume up to 50 microliter. It is advisable that pay close attention to the volumes used for each reagent. Amplification buffer is used 5 microliter, DNTP is 1 microliter, primers 2.5 microliter each, and enzyme 1 to 2 units. The water volume is variable depending upon the volume of the template DNA sample. So adjust the volume of water to get maximum volume of 50 microliter. If the thermal cycler is fitted with the upper heated lid, proceed further, place these tubes in the thermal cycler, which is pre-programmed for their denaturation, annealing and extension cycle. But if the thermal cycler is not fitted with heated lid, then add 1 or 2 drops of oil in the mixer so as to avoid evaporation during the procedure. Now place these tubes or micro titer plate in the thermal cycler and amplify the DNA sample through denaturation, annealing and amplification cycle by using this curve or these time points along with the temperature. For 30 cycles, the denaturation for 30 seconds at 94 degrees centigrade, annealing for 55 degrees centigrade for 30 seconds, and extension or polymerization for 1 minute at 72 degrees centigrade. Keep in mind 1 minute for each 1000 base pair amplification. This graph shows you a temperature curve during the PCR cycle. This point from here to here is increase in temperature up to the denaturation temperature is achieved for the particular time 30 seconds. After 30 seconds, the temperature drops for annealing again for 30 seconds, and then the temperature is increased for extension 72 degrees centigrade for 1 minute. After 1 minute, the temperature again goes towards the denaturation temperature, and this cycle is continuously repeated for 30 cycles or more as you planned. Once this procedure is over, the 30 cycles have been run, the next step is to analyze your amplification products. For this purpose, take out the plates or take out the tubes, withdraw some sample like 5 to 10 microliter from the reaction mixture and from the positive or negative control, and analyze it through gel electrophoresis. The gel electrophoresis will show you the amplification is successful, it is specific. Any non-specific amplification in negative control or in the samples, that will help you to decide whether your results are true, absolutely as per your required specific gene, or there were some errors and contaminations. While doing gel electrophoresis, always use a DNA marker so that you can determine the size of the amplified DNA sample, and you can be sure that this amplified DNA is the exactly similar and the same DNA which you wanted to amplify. This gel can be stained using ethidium bromide or cyber gold to visualize the DNA. In case you have used mineral oil to overlay the sample because of absence of the heating lid in the PCR thermocycler, you have to remove the oil, and it can be done by chloroform extraction.